Hi, welcome to SR Concepts. I am Ravi Verma, and in this video I'm going to discuss about the NCERT solutions of chapter number 5, that is the fundamental unit of life, from your NCERT textbook of science for class 9th. So in this video we will discuss the question answers of all the questions, the answers of all the questions given in your NCERT textbook. And first of all, we will start with the NCERT in text questions, the questions which are given within the text of your chapters. So, first question: who discovered cell and how? So, Robert Hooke discovered the cell in 1665. He observed thin slices of a cork through a microscope, and the cork was made up of the bark of the Spanish oak tree. So, when he observed with the microscope, right, certain empty compartments which resembled the honeycomb like structures were observed. To these empty compartments, Robert Hooke given the name cell. Now, the next question: why is cell called the structural and functional unit of life? So, dear students, as you know, that body of all the organisms is made up of cells. So, this is why it is called the structural unit of life, because cells combine together to make tissues make organs, and then organ system. So this is why the cell is called structural unit of life. Now, the cell also performs the fundamental functions, life processes such as respiration, digestion, excretion in all the unicellular and multicellular organisms. So because the cell performs functions, this is why it is called functional unit of life, and combinedly it can be called the structural and functional unit of life. Next question: how do substances like carbon dioxide and water move in and out of the cell? So carbon dioxide moves by the process of diffusion. You know that carbon dioxide is produced and accumulated inside the cell during cellular respiration process. So when cellular respiration is going on inside the cell, carbon dioxide is being produced inside the cell and it is accumulated in high concentration. But outside the cell, there is lower concentration, right, in the outside environment. So you know that diffusion is the tendency of substances to move from region of high concentration to region of low concentration. So because inside the cell there is high concentration, so carbon dioxide moves outside, because outside the cell there is lower concentration of carbon dioxide. Now we talk about water, H2O, right? This is H2O, like this. So H2O, water diffuses by the process of osmosis. So diffusion of water is called osmosis across the cell membrane, right? So osmosis is the is the diffusion of water from region of high concentration to region of low concentration through a selectively or semi-permeable membrane until the equilibrium is reached. Equilibrium reached means when the amount of water becomes equal on both the sides of the selectively permeable membrane. Now the question number 4: why is plasma membrane called selectively permeable membrane? Because it selects and allows only certain selected means entry and exit of only selected substances through the cell, and restricts others to permeate it, and this is why the cell is called selectively permeable membrane. So it will allow only certain substances to pass through it. This is why it is called selectively permeable membrane. Now the difference between prokaryotic and eukaryotic cell: you know the size of prokaryotic cell is 1 to 10 micrometer, but eukaryotic 5 to 100 micrometer. 1 micrometer is 10 to the power minus 6. Nuclear region is poorly defined without nuclear membrane, and this is called nucleoid. While the nuclear region or nucleus is well defined because it is surrounded by nuclear membrane. Membrane bound cell organelles absent in prokaryotic cell, but membrane bound cell organelles like chloroplast, mitochondria, plastids, right, and endoplasmic reticulum, they all are present in eukaryotic cell. Chromosome is single circular DNA molecule. Single circular chromosome is present in prokaryotic cells, but multiple linear chromosomes are present in eukaryotic cells. Examples are bacteria and mycoplasma, and plants, animal, fungi, and protozoa. These are the examples of eukaryotic organisms which have eukaryotic cells. Question number 6. Can you name two organelles who have their own genetic material? Yes. Mitochondria and plastids have their own genetic material, and they are called semi-autonomous organelles because they contain genes encoding some of their proteins in their own DNA. However, for rest of the proteins which they require, they depend on nuclear DNA. So for certain proteins they have their own genes, but most of the proteins which they require, they are encoded by the genes present in the nucleus of the cell. Now, if the organization of the cell is destroyed due to some physical or chemical influence, what will happen? In that case, it will not be able to perform its functions and it will die. Next question. Why are lysosomes known as suicidal bags? Because during the cellular metabolism disturbance, when there is a disturbance in the cell's metabolism, right? Beyond a certain limit, then lysosomes may burst and their enzymes digest their own cell, right? This is why the cell is called a suicidal bag of the cell. So when the cell's metabolism is disturbed, lysosomes may burst and destroy or digest whole the cell, and this is why the lysosome is called suicidal bag. Now, where are proteins synthesized inside the cell? The proteins are synthesized on particles known as ribosomes, and this is why the ribosomes are called protein factories of the cell. But ribosome particles are found attached to the rough endoplasmic reticulum's surface, and also they are found in free state inside the cytoplasm. So ribosomes are found in two states. One is attached to the surface of the endoplasmic reticulum, and the second is they are found in freely in the cytoplasm. Also they are called cytosolic ribosomes. Now we talk about exercise questions. Then first question is comparison between the plant and animal cell. This is the plant cell, this is the animal cell. So if we talk about features, cell wall is present in plant cell but absent in animal cell. Plastids present in plant cell, absent in animal cell. But if we talk about centrioles and centrosome, they are absent in plant cell but present in animal cell. And the function of centriole and centrosome is during the formation of spindle during cell division. So centrosome or centriole help in making spindle formation, right, during the cell division. Now we talk about vacuoles, right? Then a few, only one or two large vacuoles are found in plant cells, but in animal cells they may be absent, or if they are present they are very small, and there are many vacuoles in the animal cell. If we talk about size, generally plant cells are relatively larger but animal cells are relatively smaller, right? Golgi apparatus, many simple units called dictyosomes are present, right? But if we talk about Golgi apparatus in animal cells, then a single complex and prominent Golgi apparatus is present in animal cells. Question number 2. How is prokaryotic cell different from eukaryotic cell? This we have already discussed in text questions. Question number 3. What would happen if the plasma membrane ruptures and breaks? So, if the plasma membrane breaks or ruptures, then you know that the molecules will move in and out of the cell freely, and the exchange of the materials will not be there. The mechanical barrier of the cell will be broken down, so there will be no exchange of material in a controlled way. So in this way there will be free movement of molecules, free movement of substances in and out of the cell, and probably the cytoplasm, not probably but certainly cytoplasm will move out of the cell, and in this way you know that the cell will die. So cell will die. Now what would happen to the life of a cell if there is no Golgi apparatus? You know Golgi apparatus performs a few certain functions, very important functions inside the cell. So if there are no Golgi apparatus, then those functions will not be performed inside the cell, right? For example, modification and finishing of proteins and lipids formed on ER will not be done, because this is the function of Golgi apparatus. So the proteins and lipids which are formed on endoplasmic reticulum, they are sent to Golgi apparatus for giving a final form to them, means finishing of them, right? Modification of them. So this will not be happened, right? Next, packaging and dispatch of the materials to their target locations will not be done, means the materials which have been given final form and function inside the Golgi apparatus will not be packaged and will not be transported to their target locations. Now, cell will not be able to build the materials for cell wall. Here it is written cell wall and cell membrane. So the materials for cell wall and cell membrane will not be formed in that case when the Golgi apparatus is not present inside the cell. Next is lysosomes will not be formed. As you know that lysosomes are formed from Golgi apparatus. So lysosomes will not be formed, and the function which lysosomes perform will not be performed. For example, lysosome perform that they digest the worn out cells, cell organelles, and they digest the materials which are engulfed by endocytosis, right? So these materials will not be digested if lysosomes are not there. Okay, so what would happen, that worn out cell organelles will accumulate, and any material which is engulfed by endocytosis inside the cell will not be digested inside the cell, because lysosomes are not being produced, and they are not being produced because there is no Golgi apparatus inside the cell. Now question number 5. Which organelle is known as powerhouse of the cell? Mitochondria is known as powerhouse of the cell. Why? Because it releases energy from food molecules during the process of cellular respiration and converts it into chemical energy in the form of ATP molecules, which are known as adenosine triphosphate molecules. Right? These ATP molecules can give energy to numerous essential chemical activities of the life, right? And hence ATP molecules are called energy currency of the cells. Okay, so you got that that mitochondria performs cellular respiration, produces ATP molecules, which are the energy currencies of the cell. Now, where do lipids and proteins constituting the cell membrane are synthesized inside the cell? So lipids are produced on smooth endoplasmic reticulum, proteins synthesized on rough endoplasmic reticulum, especially those proteins which are used for cell membrane. Okay, now how does an amoeba obtain food? By the process of endocytosis, right? Plasma membrane of amoeba makes finger like foldings or outgrowths called pseudopodia. These finger like projections enclose the food particles and engulf it as a food vacuole, and finally lysosomes come and start digesting this food. So the process is called endocytosis. Next question is, what is osmosis? The process of movement of water molecules from region of high concentration to region of low concentration through semi-permeable membrane is called osmosis. Next question is, carry out the following osmosis experiments. So dear students, this experiment need to be performed practically, so we will not discuss it here. Now the last question is, which type of cell division is required for growth and repair of the body, and which type is involved in the formation of gametes? So dear students, in the previous video we have discussed about mitosis and meiosis. You know, mitosis is required for growth and repair of the cell, and meiosis, cell division, is done for the formation of gametes. So mitosis for growth and repair, meiosis for gametogenesis or formation of gametes. Thank you for watching this video. Please note down these question answers. I have compiled these questions especially for you. Thank you. And if you like the video, please subscribe to the channel, share it if you require, if you need that it should be shared. Thank you.